So, good morning. My name is Jefferson G. Puntawe, and I'm going to discuss what are the mutations and gene regulations. So, let's start. In this picture, we are looking at the DNA, genes, nucleotides, alleles, and mutations. So, let's start. DNA is the chemical name for this long, stringy stuff that sits inside of cell. Next. Alleles are different versions of the same genes. There are small differences in their nucleotides sequence. Genes. Genes are segments of DNA. Most genes code for proteins. Nucleotides. Nucleotides are the building blocks that make up DNA. And lastly is the mutation. Mutations are DNA copying errors when they happen in egg and sperm. They are the source of the new alleles that can pass through offspring. So, that is the intro. Mutation. Mutations are changes in genetic sequence, and they are main cause of diversity among organisms. So, mutations can be inherited. Parents that have mutation can pass them to their offspring. Germline mutations are present in every cell of an individual, including the egg and sperm used in the productions of offspring. De novo new mutations occurs by chance in one of a few eggs or sperms or just fertilizations and not present in every cell of a parent. This explains situations where a child has genetic disorder that unseen in the family history. So, mutations acquired gas environmental agent called mutagens can alter DNA. An example of a common mutagens are the UV wavelength in the sunlight associated with the skin scale. Acquired mutations are typically not passed to offspring but can be if they alter DNA sequence in egg or sperm. So we have the types of mutation. So first is the mutation, point mutation, a change in a single nucleotide. So we're at the first picture, it is the original sequence, and the other one is the point mutation. So you're at this one, and this one, the difference between the point. Okay, next. Deletions. Change the number of DNA based from removing a piece of DNA. So, this is the before deletion. So, meron siyang blue color. And then, if after the deletions, it has parang, they were going to remove some parts. Next, insertion. One or more bases are inserted into the original DNA base sequence. This is sometimes called a frameshift mutations because it shifts the codon reading frame. So, it has 4 chromosomes and an area being inserted to chromosomes 20. And then, after the insertions, the chromosome 4 becomes the chromosomes 20. Next, duplication. Consists of a piece of DNA that is abnormally copied one or a time. This type of mutation may alter the functions of the resulting proteins. So, duplication mutations. So, we have the original chromosomes and other after the chromosomes. So, it duplicates the chromosomes na yung sections of DNA is duplicated na. Next, inversions. So, it is a type of mutations where a sequence of nucleotides in a DNA is reversed. So, reverse, from the word reverse, it's exchange lang. So, this one, you're looking at original chromosomes and the mutated chromosomes. They were just exchanged. So, gene regulations include a wide range of mechanisms that are used by cells to increase or decrease the production of specific gene products, so protein or RNA. Gene regulations can occur at various steps. The amount of product depends on, first is the rate of mRNA synthesis or the transcriptions. It is the first step in the gene regulation. It involves copying a gene's DNA sequence to make an RNA molecule. mRNA degradations. Each mRNA differs the rate at which they are translated into proteins, and it is the stability of mRNA molecule. The longer lasting of mRNA molecule is the more protein product that can be transcribed from the mRNA sequence. Protein synthesis translations. The process by which of a protein is synthesized from the information contained in a molecule of a messenger or mRNA, then a transfer of RNA or tRNA molecule carrying the amino acids methionine binds to what is called the start codon of the mRNA sequence. So examples of gene regulations, enzyme inductions is a process in which molecule, drugs, induce, initiate, or enhance the expressions of an enzyme. The La operant is the interesting example of how genes expression can be regulated. And also, viruses, despite having only a few genes, possess mechanisms to regulate their gene expressions, typically into an early and late phases. Using colony system regulates by anti-terminators or splicing modulators or HIV. So, end of topic. Thank you.